everyone, and welcome to the webinar. My name is Margaret Beer, and the topic of today's webinar is bioactive contaminants in park waters. We're fortunate to have Rebecca Weisinger from the Northern Colorado Plateau Network on hand, and also Dave Vandermeulen, who's also with INM, who will be presenting today. Couple little details. One is the webinar is being recorded, and I will send out a link to the recording to everyone who registered for the webinar probably sometime later today. It will be posted on YouTube as an unlisted video, so anyone, whether they're Park Service or non-NPS, will be able to access and view the webinar. One other thing is, if you have questions, please go ahead and type your question into the question panel that you should see as part of the GoToWebinar software. We will hold questions until the very end, but when you have a question, just go ahead and type it in and submit it, and then we'll run through all the questions at the very end. So I think with that little introduction, I will hand things off to Rebecca. Rebecca, take it away.
Okay, thanks, Margaret. As Margaret mentioned, I work for the Northern Colorado Plateau Network, and the networks are part of the Inventory and Monitoring Division of the National Park Service. And we are supposed to give scientific information to park managers so that they can use that information in their policy and decision-making process. So today, David and I are going to talk to you about bioactive contaminants in national park waters, and we're going to use two case studies, one from the Great Lakes and one from the Colorado Plateau. So first of all, what do I mean when I say bioactive contaminants? Well, for this project, it's sort of four categories of chemicals: pharmaceuticals, personal care products, wastewater indicators, and pesticides. They're also known as contaminants of emerging concern or emerging contaminants, and they have a few things in common with each other. The first thing is that they are primarily human sourced. That means that they are synthetic chemicals that wouldn't be in the environment unless people put them there. There are a couple of analytes that are an exception to this that do have natural sources but are good indicators of waste, but for the most part, they are synthetic. The second thing they have in common is that they are mostly unregulated. And what that means is that there's not any kind of benchmark or water quality standard from a state or from the EPA that says you're not allowed to have more than this in the waters around us. And generally speaking, you can't have a benchmark or a standard unless you know what a safe concentration is. So for the most part, these aren't regulated because we know they're safe, but because we don't know what that safe concentration is. They're also rapidly increasing in the environment, but with little ecological knowledge about what's happening once they're in the environment. And the number of these chemicals is also rapidly increasing. It's in the thousands now. And finally, many of them are designed to be biologically active. So if you have a prescription drug that is supposed to affect your body, or if you're using an herbicide that is supposed to affect plants. So when we think about where these kinds of chemicals might be coming from, of course, when we think about pollution, we have these pictures that immediately pop up in our heads about factories and large-scale agriculture and things like that. But many of these contaminants also come from more unusual sources. So these are household products, cosmetics, fabrics, things like that don't necessarily come from one big source like a power plant or a wastewater treatment plant, but from dispersed sources. And another way that these chemicals can end up in national park waters is through atmospheric transport. So the chemicals are released into the environment. They generally latch on to very tiny particles that can get blown up into the atmosphere, transported long distances, and then rain down in more remote regions. So the Environmental Protection Agency and the National Park Service was really interested to see if the bioactive bioactive contaminants we're looking for can be found in these more remote places in national parks. So first of all, how are we looking for them? We started a screening program with the EPA Region 8 in 2010. There's over 250 compounds in the analytical suite. And I will say that both the Northern Colorado Plateau Network and the Great Lakes Network have used the information from the screening program to actually develop follow-up projects with the USGS and for the Northern Colorado Plateau Network, also with the Office of Research and Development for the EPA. So the methods are pretty simple. These are just grab samples. For all of the sites, we've been able to do the pharmaceuticals, pesticides, and personal care products, and that's over 200 analytes. And then for a subset of sites, we also were able to sample for wastewater indicators, and that was about 50 analytes. Due to lab capacity limitations, they aren't doing the wastewater indicator suite anymore, so that's why that one has dropped off. The EPA Region 8 lab in Golden, Colorado analyzes the samples, and the reporting limits are typically between 10 and 50 nanograms per liter. And the project has standard field and lab QA/QC, so blanks and replicates and clean sampling techniques. You do have to be a little careful about not contaminating samples because we carry a lot of these chemicals on our body. And one of the nice things from the network perspective is that the lab sends the data to the Water Resources Division of the Park Service, and they handle the data management to post the data to the National EPA STORET database. And then we get the data back and we do some analysis and reporting. So that's sort of a broad overview of the project that's available through EPA Region 8 for networks and national park units. And now I want to talk more specifically about what we've done in the Northern Colorado Plateau Network. And I'm just going to look at a subset of our data from 2012 through 2016. So many of you may not know where the Colorado Plateau is. It's sort of a semi-arid plateau in the Four Corners region of the United States. So Utah, Colorado, Arizona, and New Mexico. And my network works with the northern end of that, primarily in Utah, but also into Colorado. And we do have a sister network in the south that has other park units also. So this area is primarily naturally landscaped. There's no major urban centers on the plateau. Much of the land is federally owned. There's no significant large-scale agriculture, sort of dispersed livestock grazing and some alfalfa ranching. But there's not a lot of point sources of pollution on the plateau. It's also a semi-arid region, so the water that we have tends to be either larger rivers that are snowmelt driven, and these are things like the Colorado River and the Green River, or very localized and isolated water sources like springs, and we do have some cave pools as well. We do have quite a few park units on the Colorado Plateau. It's a very beautiful area with a lot of protected areas. And our parks include some world-renowned parks like Zion, Bryce Canyon, and Arches that receive well over 1 million visitors each year. And visitation is rapidly increasing at these parks. So over the course of 5 years, we sampled 21 locations in or near 8 of those National Park Service units. We sampled at varying intervals, and because we're really interested in screening and seeing where these types of chemicals are occurring, we really tried to pick a variety of water types to sample as well. So how common are contaminants? Overall, our detections are quite low. So only one contaminant was found in more than 1/3 of our Park Service samples, and that was the insect repellent DEET. And just to put that in context, during the same period for some samples in the Denver urban area, they had over 50 contaminants that were found in more than 1/3 of one samples. But the contaminants are out there. So for sites that had 4 or more visits during this time period, over half of them had a pharmaceutical, prescription, personal care product, or wastewater indicator present on more than half of visits. And what that means is that not necessarily we wouldn't see the same thing each time, but we would go out and we would find one thing one time, and then we'd go and another thing would pop up another time. So this is really consistent with kind of unique single introductions of contaminants in the system, which potentially could go back to unique individual park visitors visiting the system. And what did we find? This is the top 10 of our most frequently detected chemicals. And high on the list were personal care products that we probably all recognize, DEET and caffeine. We also commonly found flame retardants. And these are the kinds of flame retardants that are found in clothing and other household products. They are not wildland firefighting flame retardants. We did have a couple of pharmaceuticals that commonly occurred and one herbicide. So given that we are finding these chemicals out there, do we need to be concerned about it? So the overall story with this is that the detected concentrations we found are unlikely to cause biological activity in vertebrates. There's sort of a lot of information that goes behind that statement. So the first thing I want to talk about are nanograms per liter. So these are reported in nanograms per liter, and that's equivalent to 1 part per trillion, which is very difficult for me to visualize. So if you can imagine that you're standing in the middle of Zion National Park, or about 250 square miles, 1 part per trillion would be 1 square inch in front of you. And most of the detections that we had in our data set are in the tens of nanograms per liter. So this is very tiny concentrations that are being detected in our park waters. However, that doesn't necessarily get us off the hook, because these are supposed to be biologically active components. So, you know, maybe we need to be concerned about that. So when you look at the literature, most of the compounds don't have any information, but there are a few out there, things like metformin and BPA. And as you look for effect sizes, most of the time they're in the thousands of nanograms per liter. So from the literature for the compounds that have been studied both in the lab and environmentally, we seem to be below that. But that wasn't really good enough for me to feel comfortable with these data, because most of the compounds don't really have any information at all. And so to kind of get at this, we worked with the EPA's Office of Research and Development, and specifically Brett Blackwell, to compare the maximum concentrations in our data set to the ToxCast database. Now, unlike benchmarks and a lot of toxicity information, which focuses on basically when organisms start to die, so mortality level effects, ToxCast is a bioassay of a huge variety of cellular endpoints. And they expose these cellular endpoints to different concentrations of many, many chemicals, and started to look for cellular activity. So it doesn't only include mortality, but it could show things like endocrine disruption or neurological endpoints or cardiovascular endpoints. So in comparison to toxicity, it should be a much more conservative estimate of the risk of biological activity in organisms. So when we looked at the maximum concentrations we detected anywhere at any site, none of them exceeded the threshold for expected biological activity based on ToxCast. So that's good news, but there are a couple of caveats to that. One is that the cell lines are all vertebrate cell lines, so it probably doesn't give a good estimate of risk for invertebrates or microbes or algae, things like that. And the second caveat is that we did have some hormones in our analytical suite, but for the hormones, the reporting limit actually was a little bit high. So we had a lot of non-detects, but it's possible the hormones occurred at lower concentrations that we couldn't detect that could still be biologically active. So what sites are vulnerable to contamination? As you'll recall, we tried to pick a variety of sites from big rivers to really isolated springs. And when we compared those two categories of waters, we didn't really see any differences in the average detection frequency or the median detection concentration. So as a blanket statement, it seemed like things were kind of getting out all over the place. But when we limited it to looking at sites that had chronic contaminants, so in contrast to this, the sporadic, like one-off kind of contamination, the sites that we consistently found the same chemical over time, there were a couple of patterns that came out. And the first was that when we had low flow, poorly mixed systems, we seemed to be more likely to detect this chronic contamination. And the first example I have for that is with a pesticide. On the left, you can see Courthouse Wash in Arches National Park. It's a spatially intermittent stream, so it has sections that are perennial, but it has sections that are dry in between, and it's not connected to upstream sources by any kind of flow. And the sampling there, we detected triclopyr on more than 30% of visits. And triclopyr is an herbicide that is used for removal of an invasive exotic shrub called tamarisk. Now, when you compare that to the Fremont River, which is next to the historic orchards at Capitol Reef National Park, which you can see on the right, we had no chronic contaminants in the Fremont River, although you can see that the orchards are a highly managed system. Another place where we saw this pattern was for personal care products in cave pools at Timpanogos Cave National Monument. So Timpanogos Cave National Monument is located high in the watershed. It's underneath Forest Service land that is alpine and partially wilderness protected. So there's not a lot of development happening upgrading it from these pools. But there are cave tours that go through the cave where thousands of people go through every day during the recreation season. Now, they're not going into the cave pools, but the path does go directly next to, or in some cases, over the pool on a bridge. So in these cave pools, we detected DEET and caffeine on more than 30% of our samples. Now, in comparison to that, we also sampled the Narrows in Zion National Park. This site also gets thousands of visitors every day, and in this case, they're directly contacting the water. They often go wading in the water. And for the Narrows samples, we did not have any chronic contaminants. Another type of system where we found this sort of chronic contamination are in systems that were influenced by wastewater effluent. And in this case, generally we found pharmaceuticals on more than 30% of visits. So these are places like the Colorado River upstream of Canyonlands National Park and the Yampa River in Dinosaur National Monument. Most often, the pharmaceuticals we found were metformin and gabapentin. I'm not going to talk about them much because Dave talks about them later, but I will say I don't think it's because these are the most prescribed prescription drugs out there, but that they seem to perhaps persist a little longer and so show up more often. So to kind of sum up these pathways and patterns, these pseudo-persistent pharmaceuticals seem to indicate a dilute wastewater effluent signal. Low flow and poorly mixed waters seem to be at a higher risk for persistent or chronic contamination. And I think that's just because they can't flush things out that easily. Direct and indirect visitor contact likely introduces contaminants, and park management, including pesticide use, can also introduce contaminants. So what can managers do? Well, you can encourage visitors to eliminate liquid and solid waste at least 200 feet from water sources when possible. And I know most parks have, backcountry regulations that stress digging cat holes and placement of cat holes, but less often do we talk about liquid waste. So that's something we could potentially add to backcountry guidelines and regulations. In places where we have low flow systems like arid land springs and cave pools, we can discourage visitor contact with that water. In the Northern Colorado Plateau Network, the message we've put out is that, these systems, because we're in a dry area, these systems are also very important for wildlife. So let's just leave those little pools and stuff for wildlife and keep our distance. And then another thing we can do is encourage parks and communities to update aging wastewater treatment facilities. I want to say that it's very, very difficult to completely remove pharmaceuticals and personal care products from wastewater effluent, and that's not a very realistic goal, but new wastewater treatment facilities or modern wastewater treatment facilities have a much longer processing time. They allow more time for microbial degradation, and so they can significantly reduce the concentrations in effluent. So what's the story? Where people are, chemicals follow. The low concentrations we detected shouldn't affect visitors. Dilution is still the solution to pollution. So if you have more water, these contaminants, it takes more of them to show up as a problem. Whereas if you have less water, even very tiny concentrations can persist. And improving aging wastewater treatment plants could help. So I have a bunch of people that I need to acknowledge for this. The bulk of the presentation today is based on a manuscript that came out in Science of the Total Environment this spring, and it had both EPA and USGS co-authors. And I wanted to show you, I have a couple of links in here. The first is to a brief that we put out on this manuscript. And the second is to the abstract of the manuscript itself. The manuscript is behind a paywall, so if you want a copy, just contact me. And we're going to hold off questions on my portion until the very end of the webinar. So I'm going to hand it over to Dave now so that he can talk about what they've found in a very different system from the Colorado Plateau.
Hey, Dave, I'm passing it over to you.
Okay. Let's see here. All right, can you see my screen?
Looks good.
Okay, and I assume everybody can hear me. Well, like Rebecca said, my name is David Vandermeulen, and I'm with the Great Lakes Inventory and Monitoring Network. And right up front, I'd like to acknowledge a key collaborator, Sarah Elliott. She works for USGS out of the Mountains View, Minnesota office. She's been really important in helping me compile and synthesize and analyze data. Well, just as an overview, I want to start out by introducing the Great Lakes I&M network, similar to what Rebecca did for her network. I'll go over some general findings over a 5-year time span there, 2013 to 2017. I'll pick out a couple of noteworthy examples to talk about and go over some future work, and then conclude my portion of this talk. So you can see here, on the left there is a map of all the I&M networks. The Great Lakes network is located aptly next to the Great Lakes, and off to the right you'll see a map of the 9 national parks in the Great Lakes network. What I'll be talking about results from the parks shown in green. We have one park, St. Croix National Scenic Riverway, where we're sampling in 2018. And I'll be talking about that at some point in the future. So to talk a little bit about our sampling, so like I just mentioned, 8 out of the 9 parks were sampled from 2013 to 2017. The picture of the sample bottles there off on the right. The number of sites per park ranged anywhere from 6 to 19. And it covered many different types of sites. You know, for example, the Mississippi River that flows to the Mississippi National River and Recreation Area, inland lakes at 4 or 5 of the parks, and then streams and ditches at some others. And we even sampled one of the Great Lakes, Lake Superior, at Apostle Islands National Lakeshore. And I do want to mention, and I suspect this was the case for Rebecca as well, that the sites were not randomly selected. At least in the Great Lakes, the sites mostly overlapped where monitoring was already scheduled, usually routine water quality monitoring. We wouldn't have had the ability to develop independent sampling for this project, independent of our other monitoring for all the parks. We did do that for a few parks though. 2 of the parks, Indiana Dunes National Lakeshore and Mississippi National River and Recreation Area, we sampled for a couple of years, whereas the others we just sampled a single year. In any given year, which the sampling window is April through November, we would visit each site 1 to 3 times. And like Rebecca mentioned, each of the samples was analyzed for over 200 different contaminants of emerging concern. So the big picture across all 5 of those years was that we collected 242 field samples. There were a number of quality control samples, both field and lab, associated with them. So out of those 242 field samples, we have the potential, at least, for over 50,000 unique results. But out of that 51,372 potential results, we actually detected contaminants in just over 1,200 of them. So about 2.4% of the time. So if you look across all the samples, we're averaging roughly 5 contaminants per sample. But how does that break down by park? This next figure shows, again, a map of the Great Lakes parks on the top. And if you look down below, our 2, what I'll call more urban parks, Mississippi National River and Recreation Area right here in the vicinity of the Twin Cities, Indiana Dunes National Lakeshore, which is just east of Chicago. Those are urban parks, and we had many, many more detections of pharmaceuticals and personal care products and even pesticides at those parks in comparison to all the other parks where we sampled, which are more, much more rural. So urban areas, many more detections of contaminants. And this is in line with the example that Rebecca gave from Denver as well, where they were detecting approximately 50 contaminants per sample there. And what were we detecting? Here I'm listing the top 7 things that we would detect when we would get detections. And this list is a little bit different than what Rebecca had. But most of the chemicals on this list are herbicides. The exception to that would be this gabapentin, and which is an anti-seizure pain medication, and metformin, which is taken to treat diabetes. So most of the time we're getting herbicides or DEET, insecticide, but we do have these 2 other pharmaceutical types in our to be detected. But just remember that what I just mentioned is that sampling effort was not equal across the parks. And I circled gabapentin here in red because I'm going to return to that in a couple slides and talk a little bit more about that. Well, I guess I'll talk about it right now. This is one that caught my attention because when we first started doing the sampling in April of 2013, I was the one collecting the samples on the Mississippi River, and we were getting data back. And the levels of this gabapentin were quite a bit higher than other things that we were detecting in those river samples. Gabapentin is an anti-seizure drug that relieves pain and anxiety. It's, at least historically, widely perceived as safe for people, and it's actually given to pets as well. Through a little bit of internet research, I've come to find out that gabapentin taken in conjunction with opioids can increase the opioid high. And actually, if you take too much of it in conjunction with opioids, it does increase the risk of opioid-related deaths. So what really caught my attention was the high levels that we were getting in the river relative to the other contaminants. And down at the very bottom there, you'll see that we did detect it fairly frequently at Apostle Islands National Lakeshore, about 1/3 of the time, Lake Superior samples, and also in the streams and ditches at Indiana Dunes National Lakeshore, about 2/3 of the time. But for all the sampling work we did on the Mississippi River, we detected this in every single sample. The figure on the left shows the 4 river sites where I did the sampling. The Elk River, Minnesota is the furthest upstream. So going left to right, we're going downstream, and you can see an obvious increase in the chemical from upstream to downstream. And there also potentially is this sort of potentially a seasonal effect there, where much higher levels in the spring, and then a drop off into the summer. And I did a little bit of data exploration here to try to figure out what was going on, and that's shown off on the right. The figure on the top right shows gabapentin, and on the y-axis as potentially a function of river discharge, but there doesn't seem to be much of a relationship there. Figure on the bottom right shows gabapentin potentially as a function of water temperature, and you see that there's a possibility of a connection there with the concentration of gabapentin being much higher in colder water compared to warmer water. And I don't have an answer to this, it's just a little bit of exploration that I did there. Now I want to take a few minutes and just focus on Indiana Dunes National Lakeshore. Like I mentioned earlier, it's just east of Chicago on the southern end of Lake Michigan. Here's Gary, Indiana right here. And the park is shown outlined in purple, and all these boxes with numbers next to them are sample sites. So there's 19 sample sites at the park. One other noteworthy thing to take note of here is that there's a wastewater treatment plant shown there in the red circle, and water generally flows from east to west through the Calumet River, and then it's also flows generally towards Lake Michigan in this area of the park from these ditches. One thing to take note of is that the park is sort of an oasis of green in the middle of a highly developed area along the shoreline. These are heavy industry, steel mills here and here. There's Michigan City. And then off to the southeast is a big agricultural area. So the park is sort of this green oasis here. And the sites are both in the agricultural area as well as downstream of the treatment plant and in the more urban area. And if you just take a look at the top panel here, I'm just focusing on pharmaceuticals and personal care products. Again, here's where the wastewater treatment plant is located, and the sites shown here in bigger circles and darker color, they have the highest number of detections downstream of the wastewater treatment plant and more generally in this more urban area over here in the park. Whereas up in the agriculture area, not as many detections of pharmaceuticals and personal care products. But in contrast to that, if you look at the panel on the bottom, there we're just focusing on pesticides. You can see that sort of in this more agricultural area to the southeast, we're getting, generally speaking, a higher number of detections of pesticides in this area. So we can just use this information in a general way to kind of break it out between areas with higher numbers of pesticides and areas with higher detections of personal care products and pharmaceuticals. One particular herbicide that caught my attention at Indiana Dunes was this herbicide called simazine. It's like a white granular product that's put on fields, and it's long acting. It's active for 2 to 7 months, and it basically suppresses weeds. It's some sort of a granular product that's not supposed to be very soluble in water. The reason why it caught my attention is that this was the only park where we detected simazine. And when we look at where most of the detections occurred in these red circles, most of them occurred, in this agricultural area. You did get some downstream here, but generally speaking, more of the detections are up in the agriculture area of the park. The next example is this ecgonine methyl ester. It's actually a metabolite of cocaine, and when we look at where we detect that, it's mostly downstream of the wastewater treatment plant, and then over here in these areas that border, this is more of an urban area that borders residential development, and not so much of that in the agricultural area. So just two items of interest from Indiana Dunes National Lakeshore. But when we take a look at what I'll call our rural or wilderness parks, shown in the big red circle there, we had many samples that we sent in that came back with 0 detections. But when we did detect chemicals, most of the time it was atrazine and DEET. And I guess like Rebecca, I'm going to presume that most of the DEET was coming from park visitors or potentially park staff, whereas the atrazine most likely was through dust getting picked up off of farm fields in the Corn Belt to the west and south of where all of our parks are. So, this slide, a lot of it mirrors what Rebecca was saying about potential impacts to human and ecological health. at least in the Great Lakes, I guess like Rebecca found, when detected, levels of the chemicals are almost always orders of magnitude less than published standards or reference values. There was one exception of a chemical called hydrochlorothiazide. It's a diuretic used to treat high blood pressure, where we had a few occasions where detected concentrations were just above a Minnesota Department of Health screening value, but that value is very conservative at 40 parts per trillion. So I just would mention the caveat similar to Rebecca, that standards exist for relatively few chemicals, and it really limits your ability to interpret overall risk. Standards also don't account for the fact that all these things exist, these chemicals, when they're there, exist together in mixtures. And many studies have shown adverse biological effects on organisms exposed to fairly low levels of these chemicals, including those with standards. So in terms of what we're doing this year, like I mentioned earlier, we're sampling at St. Croix National Scenic Riverway, six river sites. We also returned to pictured rocks where we had sampled inland lakes before. We're now sampling 4 streams in an effort to try to determine whether there's any visitor impact. We're doing upstream and downstream sampling. In 2019, at least the Great Lakes will be at a point where we need to make a decision whether or not we want to go back and resample any of the parks we sampled earlier. And there's also been some preliminary discussion about what a nationwide roll-up of this data would look like. There are other INM networks besides Northern Colorado Plateau and the Great Lakes who are participating in this, at least 4 or 5 others. And so we're just now starting the conversation of what the utility of a nationwide roll-up would look like. Like Rebecca, I've got a couple of publications here that I'd be definitely willing to share. Sarah Elliott and I also published in Science of the Total Environment. A few years ago, I published a sort of a summary report on earlier findings, and we also have one in the works that should be published sometime in the next month or 2. So far, I've given a different iteration of this talk at a different meeting, and then, of course, this webinar. And for those of you who are interested in participating in this project in the future, I would recommend contacting Karenza King at WRD, because every year in the late winter, we start to negotiate who will be able to get samples and when and how many. So just to summarize, by 2018, we'll have baseline data for a variety of surface waters across all the 9 parks in the Great Lakes network. Many more chemicals are detected at urban parks and or those exposed to wastewater treatment plant effluent or those agricultural areas. Atrazine and DEET seem to be pretty ubiquitous, and they're found even in remote locations, even though they're at pretty low levels. When they're detected, the contaminants are nearly always well below published standards or reference values, but just know that information on those are lacking. And so I'm acknowledging many of the same people that Rebecca acknowledged, but I'd also like to specifically mention staff at the EPA Region 8 Analytical Lab. Over the years, they've analyzed thousands, or over 1,000, well over 1,000 samples, and it's free. They do it all in kind. I'm guessing that if the Park Service actually had to pay for those analytical costs, we'd already be well north of $1 million. And so with that, Rebecca and I would be happy to take any questions that you have.
Okay, Rebecca, any need to transfer back over to you for as a presenter?
No, I think we can stay with Dave's slide.
Okay, okay. Again, type in a question if you want to ask Dave or Rebecca anything. And we do have one question. I hope I pronounced this correctly. Was triclopyr being actively used in the watershed that season? And I believe that refers back to your presentation, Rebecca, possibly looking at Courthouse Wash.
Yes, and the answer to that question is yes, and it has been used for several seasons previously. So tamarisk is a very common invasive shrub along riparian systems on the Colorado Plateau, and it is being actively managed. It's not put out as widespread spray. What they generally do is cut the shrubs and then paint the stumps. So it's not that they were just spilling herbicide willy-nilly, just that because there's, or in my opinion, or my hypothesis is that because it's quite a low flow system, even that low amount of herbicide in the system wasn't able to get flushed out as it kind of worked towards the stream.
Okay, I don't see any more questions right now, so we'll give everyone a couple more minutes to type anything in. And while we're waiting, just a reminder that I will be sending out the link to the recording of the webinar, probably sometime later this afternoon. Okay, another question. You mentioned that currently there are few health standards for CECs, and there aren't really methods to remove CECs from wastewater. Is there any news from EPA or industry on those two fronts?
Yes. It's not possible to totally remove them, but we can dramatically decrease them. So I don't want to, say that they're, oh, throw up your hands, there's nothing we can do. We can definitely decrease the concentrations that we see. And I will say that the Office of Research and Development for the EPA is working on some really interesting and new techniques for thinking about this kind of bioactivity question, because if they can show what levels are biologically active, then we can start to have these standards where, it would be like a water quality standard for ammonia or something like that. You have to be below it under the Clean Water Act. So we're currently working with them right now. It's Dan Villeneuve's lab, and they're out of Duluth, Minnesota. And I know one of the things they're working on are skin swabs for fish, or I guess scale swabs would be the better thing to say, where they just swab the mucus off of fish and then try to see if they can detect biological activity based on those swabs so that they don't have to kill the fish to look at liver tissue and gonads and things like that. Another thing that they're working on right now is something that would use source water samples, and instead of breaking it up into each contaminant that's out there and comparing each contaminant to a toxicity level or biological activity level, they actually put the whole sample in. It's a similar kind of bioassay approach that looks at whether the sample with all of its contaminants together are biologically active. So it gets at that chemical mixture idea that Dave was talking about. And the USGS has been working on some similar things with a yeast bioassay that looks for kind of estrogen activity. So that's a little more limited than the broader scale bioassay approach that EPA is taking. And those are the ones that I'm aware of. Dave, do you know about other things?
Not so much, but I'll just add that it seems like the effort to understand at the state level, the effort to understand potential impacts and development of standards and reference values varies pretty widely. I'm most familiar with the work that's being done in the state of Minnesota, primarily by the Minnesota Department of Health, but there's other states in the Great Lakes where I'm not aware of basically any work at the state level to develop standards or reference values.
Okay, thank you. I'm not seeing any other questions. Once again, we'll give it a wait a little bit longer in case anyone's typing in. And no further questions. So any final comments, Rebecca, Dave, before we conclude?
Thanks everyone for tuning in, and if you're interested in learning more about how you can be involved, you can feel free to contact me.
All right.
Yep, I'll just echo what Rebecca said. Thanks for the invitation to give the talk.
All right, thank you both. Thanks to everyone who attended, and hope you can join us at the next webinar. Thanks again, bye-bye.
Bye-bye.